Hello, this is Renzo, and this is rehearsal talk for the talk during the symposium organized from Amir Shmuel regarding the opening of the 7 Tesla in Montreal at the MNI Brain. And before I start, I really want to thank Amir for inviting me. It's a pleasure to speak among all these big speakers, and congratulations from my side on the new fancy Terra. I mostly do laminar fMRI with blood volume fMRI. And why are layers so interesting? Layers are very relevant to bridge different spatial regimes of neuroscience, because neuroscience really bridges everything from the microscopic scale, where we have synapses and cell bodies, up to the macroscopic scale, where we have behavior, manifestations of mental disorders, and activities across different brain areas. It's only kind of this mesoscopic part here that's somewhat underrepresented. But both sides of the bridge are trying to get closer and closer to each other. For example, with new optical imaging methods, you can measure hundreds and thousands of neurons at the same time, whereas high-resolution fMRI does not only allow you to distinguish activity between different brain areas, but also you can look at the activity distributions within those brain areas. And the keystone that will bring everything together is the spatial regime of these basic neural building blocks, the cortical layers and columns in the spatial regime of a few hundred micrometers. And to get there with fMRI, I'm really willing to make compromises. So I don't really care yet if the TR is more than 3 seconds or if it's not really a whole brain method just yet. We should try first closing the bridge and then build on top of that as soon as it's stable. And this spatial regime of a few hundred micrometers here is really relevant because it gives us a new dimension in order to investigate brain systems. So you know that we have all these different brain areas that are mostly like hierarchically organized, in the sense that any given brain area right there receives feed-forward input from an area that is lower in the hierarchy, mostly into its middle cortical layers. Whereas the same brain area receives feedback input from another area that is higher in the hierarchy, mostly in upper and or deeper layers. So by knowing at which layer the activity occurs tells us a lot about the information content, more than just which area is being involved. And obviously not the entire brain is strictly hierarchically organized, but especially in the frontal lobes, there are other canonical microcircuits that might make more sense, like this input-output model. To get to these layers, however, there are 2 huge challenges. And the first big challenge is the sensitivity, that going from a conventional 3mm fMRI voxel to an 0.75mm layer fMRI voxel is a huge reduction in volume. It's a factor of 64. And that's really a big number to deal with. So there's almost no question that we need to make compromises. And even the best new shiny Terra will not completely account for a factor of 64. The second big challenge is the specificity, that going to this high resolution alone doesn't help. In fact, when you go from conventional like 3mm voxel to 2, 1 or 0.7mm resolution voxels, you still end up with these kind of fuzzy blobs. And now you just have this blob at high resolution, but you don't really gain so much. You don't see these stripy patterns that we would be interested in, the layers. And this comes from the fact that most of the oxygenation changes are coming from these large draining veins that are not strictly laminar aligned like the neurons are. So in some sense, using conventional gradient echo bold as a measure of laminar activity is like using CO2 emission mapping as a measure of industrial activity. It works very well to compare like the big structures of different countries or different districts in Canada, but as soon as you approach the spatial regime of the draining infrastructure, the whole model breaks down, obviously, and you cannot use the overall CO2 emission of a voxel like that as a measure of the activity in those buildings over here, because right now there's a lot of activity going on right here. However, most of the CO2 emission actually comes from cars that go to other buildings outside this voxel. Similarly here, most of the oxygenation changes in the superficial layers are probably referring to neural activity further upstream in the deeper layers. And this is well known since the advent of fMRI, and there are other methods proposed to be more specific. And one of my personally favorite methods is this vascular space occupancy VASO method that is believed to be more specific to the smaller blood vessels that are closer aligned to the laminar neural structures. And this VASO method is non-invasive. It basically works with a very simple model where we just assume 2 magnetization vectors, the tissue in blue and the blood in red. And we start every TR with a global 180-degree inversion pulse, which flips these magnetizations, and then they decay with their respectively different T1 relaxation. And there's one exact time point where blood in red is exactly 0. about now. And this is the time when we play out the excitation pulse and acquire our signal. So right during this time, any voxel that has all kind of signal or tissue and water compartments actually is only sensitive to everything but blood. So white here stands for no signal, because there's no red arrow here. It's just gone through zero. And then when you have an activity increase, all the vessels open up, meaning that you have reduced extravascular signal available, so your signal goes down proportional to the blood volume increase. And this really helps us with the specificity limitation. However, we still need to deal a lot with the sensitivity. And one very efficient way to increase the sensitivity is to go to higher and higher field strength. And while clinical field strength of 3 Tesla with vaso or finger tapping in a motor cortex just barely exceeds the detection threshold, here 7 Tesla is already a whole different world, where you can start to see these stripy patterns over here indicating the different layers. 9.4 Tesla might give you some extra specialized performance here. However, it also comes along with a lot of technical challenges. And Nick Weisskopf once used this car metaphor in an educational talks years ago. I think that inspired me, where he explained it with this car metaphor. So I would like to borrow this car metaphor now to also compare the different field strengths, where 3 Tesla is really the most cost-efficient way of doing things. However, 7 Tesla really buys you some extra performance, but it's more expensive, right? On the other hand, it has been out there for long enough that you can buy it industrially, and you push a button and it basically works. These systems are well understood now. And for 9.4 Tesla, you might be able to get some extra performance out of it. However, you really need to have a whole team of engineers working on these kind of prototypes, and then sometimes you get these record-breaking experiments out of it. But 7 Tesla is quite a good sweet spot between performance and applicability. Another advancement, or another way to increase the sensitivity, is to use these recent advancements in the readout. And there have been quite a lot of advancements in the last 5 years or so about readout strategies, and not only with respect to 2D SMS and with the multibanding, but also pretty much at the same time when SMS was introduced for fMRI, also 3D EPI was introduced for fMRI, which works different in the way that you don't excite simultaneously acquired slices like in a consecutive fashion, but you actually acquire the whole volume over and over again, and you just use different spatial frequency weights. And you can use the same acceleration through both phase encoding directions then. And both of these methods have a different sensitivity spectrum across voxel volumes. So while for conventional 3mm voxels, where you are in the physiological noise-dominated regime, the 2D SMS really outperforms 3D EPI. Whereas in the thermal noise-dominated regime of these sub-millimeter voxels, where we want to see these stripy patterns here, the 3D really outperforms 2D EPI. And obviously the crossing point really depends on TR, acceleration, and all kind of things. We see that for high resolution vaso, 3D really is the way to go. So based on the previous slides, we have now a good combination of methods. We use blood volume with vaso at 7 Tesla with 3D EPI. And so now I would like to show you the first results with this combination of methods that we used, just to as a validation and method debugging, where we go into the motor cortex because it's particularly thick, and we have this very simple microcircuit of cortical input in the superficial layers and cortical spinal output in the deeper layers. And then we try to modulate these different channels with different movement and touching tasks. For example, for a tapping with touch over here, we do move the muscle, so there must be output triggers from the deeper layers. However, we also touch the finger, so there's feedback from sensory cortex coming in, or also from motor planning areas. So we actually do expect upper and deeper layers to be activated right there, resulting in 2 peaks in blue here. Whereas for the black task condition here, we ask the participants not to move their fingers. So in the deeper layers, there's no output. It even looks like a suppression of output. Whereas the upper layers still have this feedback from the touching, from the sensory cortex. So you see that for the black one, we only have input, no output. And this made us quite happy in the sense that we can nicely differentially modulate the different layers differently. However, one thing that we learned also from these data is that you really need to know where you are in your kind of topographical space along the columnar or hypercolumnar dimension. For example, here it looks all perfect, it makes sense. However, if you move a few millimeters away already, it breaks down because there you're on a different movement representation. So in order to developed this kind of methodology to also look at columns and layers at the same time. We again used a very simple model of just individual finger tapping in a motor cortex. And to show you how this looks like, I would like to show you this real data in this little animation here, where we have a motor cortex on the right, which is super thick. And on the left side here, we have the sensory cortex. This would be the central sulcus in the center. And then in order to get to these topographical representations, we use this crawler algorithm directly in the voxel space, where you kind of grow in the different surfaces directly in voxel space for black in the sensory cortex and white for the motor cortex. And then you can look at it basically in 3D, here turning it. So now we are basically looking at the central sulcus from the back of the brain, where this would be the midline of the brain and this would be the right ear. And everything in black is the sensory cortex. And then we can look at the different finger representation here. So this here we have this would be the pinky finger, ring finger, middle finger, index finger, and thumb. So in the sensory cortex, it's all well behaved. We have a somatotopical topography, where the different finger representations span across the depth of the central sulcus and are linearly aligned. I was quite surprised to see how big it is. Like almost 30% of the central sulcus seems to be busy representing the different fingers. More interesting, however, is what happens when we go to the other side. Here, for example, turning this around, where you kind of look at this cave here, that would be the hand knob on the motor cortex. And again, I'm projecting the individual fingers onto it. So here, starting with the pinky finger, ring finger, middle finger, index finger, and thumb. And you can see that the motor cortex is kind of messy in the sense that they are much, much smaller. This entire topographically aligned hand here only covers about 4 millimeters, and we have multiple representations in a mirrored fashion, which becomes maybe clearer if we look at a kind of axial-like projection here, where you see on the bottom left that we have a sensory, huge representation on the motor cortex. On the top right, we have these kind of smaller mirrored representations. And in bold, it's kind of there too, but as soon as you have a big vein, it's all messed up, and that dominates the finger, and you don't really see the smaller hand so well. And this is actually also highly consistent across participants, where you always see this huge hand on the bottom left for the sensory cortex. On the top right, we have the motor cortex representations in a mirrored fashion. And these patches that refer to either one of both hands nicely match the different patches that refer to grasping and retraction actions. So it really makes a difference in the motor cortex how you use your finger. If you make it as a fist, or if you use the other side of the muscles and kind of span your fingers apart from each other. So until now, everything that I did referred to the motor cortex. And people say that trying to understand how the brain works by looking at fMRI is like trying to understand how a truck engine works by looking at it with an infrared camera. We only roughly know what's going on and which area is consuming energy and heating up, but we don't really understand so much about the densely packed microcircuitry and if this heating is coming from input or output. Now with layer-dependent fMRI, I showed you that we can maybe go a bit beyond that, where we not only know which area is involved, but we actually approach the regime of this microcircuitry where we can say something about the content, like if we have 2 stripes for tapping or 1 stripe for touching. But the motor cortex is so easy, right? It's not any regular truck engine, but it's like the biggest engine there is. There's no cortex as thick as the motor cortex, and you don't need any fancy methodology to easily see the densely packed microcircuitry of an engine like that. For example, input and output here, even with a small field of view. So in the last 3 years, I think we really heavily tried to extend this layer-dependent fMRI methodology to also work on more regular-sized cortexes, for example, the very thin visual system. And we wanted to go there without really sacrificing the stripiness. So we want to see these dark voxels here forming a line indicating the stria of Gennari, but not only in a tiny field of view, but actually in an entire slice covering the visual system, part of an entire slab of isotropic voxels. And since we care about activity, it's not enough to just have pretty EPIs, but we actually also need more than the blobby bold, but actually also the higher specificity of vaso, even though it comes along with a higher noise level too. And as soon as we had that, we then tried to validate it following Jonathan Polymeni's approach here of directional connectivity in resting state, where we cover LGN, V1 and V5, and they send feed-forward and feedback input to V1 respectively. Visible here as resting state functional connectivity in V1, where we have a clear feed-forward peak in the middle layers for seed region LGN in green, and a clear feedback additional peak for seed region in V5. However, as soon as we validated this and we knew that the method kind of works, we tried to move beyond these very well-controlled test cases, where usually people like zoom into a specific patch of cortex and then look at layer-dependent responses for different like task conditions. And then only try to interpret differential responses, not the absolute ones. So why not do it the other way around and look at one task condition, for example, resting state, and try to distinguish and look at differential responses across the columnar domain. So again, we are assuming a canonical microcircuit, and we just assume that a feed-forward-driven column would look like that, and a feedback-driven column would look like that. And then we can take resting state data, place a seed region, and see which column is feed-forward or feedback-driven. Here, for example, when you go along, or maybe let's start here, this would be a seed region somewhere between V1 and V2, and you see that it's mostly the calcarine sulcus, the early visual areas that receive feedback input in blue, whereas most of the visual stream actually receives feed-forward input, or clearest when we go to LGN directly, where almost the entire visual system seems to receive feed-forward input. And if we move along here to something that might be V5, you can see that the early visual areas, they all receive feedback input in blue, whereas only this area, not really sure what it is, it's still further anterior and still receives feed-forward input from that seed region. And in this way, you can easily kind of map out the columnar-to-columnar hierarchy in a functional sense without knowing any predefined ROIs. Another application of layer-dependent vaso-fMRI outside the motor system is our cognitive areas, here in a study that was headed by Emily Finn in collaboration. And also in those areas, we know from the electrophysiological monkey literature that they have very specific functional microcircuits. For example, there are kind of cue cells, there are delay cells, and there are response cells that eventually trigger the motor system. And they are aligned across different cortical lamina. So Emily Finn here found an old task that should engage these layers differently by showing participants strings of letters and then cueing them to either remembering the strings of letters in the order they saw it, or sorting them according to an alphabetical order mentally in their mind, which really is believed to engage the superficial working memory layers. And then later in the task, they get random letters presented to them, and they need to do a response decision based on the position there, which then engages the deeper layers. And this simplified microcircuitry here really nicely explains all the data that we are getting. Zooming in here to a CBV-weighted EPI that has this inherent T1 contrast, you can kind of see that the output activity in this task really nicely follows the deeper layers here. And this analysis refers to 1 millimeter smoothing within layers, whereas the superficial layers, they really care about this alphabetization versus remembering. Maybe even clearer visible when we look at group results of 14 participants, where the superficial layers, here the working memory layers, they really shoot up as soon as the participants need to start to do this alphabetization task. And when they found the right order, then it goes back to baseline. Whereas the deeper output layers, they don't really care so much about this alphabetization, and they only shoot up as soon as the participant found a solution and needs to do the response. And if you want to learn more about the neuroscientific implications, about the working principles of DLPFC, check out Emily's preprint over here. Another potential application of layer-dependent fMRI is this new field in cognitive neuroscience of naturalistic tasks like movie watching. And this kind of new field is really underdeveloped for layer-dependent fMRI just yet, even though these tasks might be super beneficial for layer-dependent fMRI for multiple reasons. For example, you can use these tasks to do any kind of connectivity analysis that you would usually do for resting state without this ambiguity of physiological to neural-driven noises or fluctuations. And maybe even more importantly, you can show participants with limits the same movie multiple times and then average, which really helps for our noise-driven submillimeter data. Or you can even show the movies multiple times and then cover different slabs of the brain and then do some retrospective combination to do some hypothetical synthetic whole brain connectivity analysis. So to start learning from these tasks and then what's possible, we are using these clips that are also used for the 70 release of the HCP data, meaning that we already have about 100 participants and really know what to expect, which kind of signal traces across the different brain areas. And then acquiring data with isotropic 8mm slab vaso. Again, the vaso helps to remove vascular noise and distinguish between vascular and neural-driven fluctuations. The kind of questions that we can try to address with these setups are referring to better understanding the source of individual differences. So here, for example, I'm looking at the task positive network during these movies, and you can see that most participants here, separated in 3 groups, have very consistent signal traces. However, there are still time points that are more stable compared to time points that are less stable. For example, when people look at these wave beach scenes, the entire brain seems to do the same thing almost always. Whereas, for example, for kissing scene, it's not so much. And then we can zoom into these time frames, look at the layer-dependent fMRI, high-resolution data, and see where this is coming from. And we are seeing that this mostly comes from the superficial layers. The deeper layers, they are really doing there's the same thing across all participants, which makes a lot of sense because all the participants see the same movie. So the feed-forward input from the eyes must be the same. The early visual areas extract the same low-level features. Only somewhere along the cortical hierarchy, these individual differences come in as feedback input here in the upper layers. Other questions that we might be able to address with these setups are regarding the directional connectivity or hubness of different areas within already described networks. For example, the default mode network, where we can again use the averaged signal traces from the 100 participants. We know where the default mode network should be. We extract the signal traces over here and then use these signal traces as regressors for our high resolution data where participants use watch the same movies. First to extract the default mode network and then look at it across cortical depth, where you can see that different areas look differently. For example, the IPC or PFC really have different profiles. For example, both of them, they have this huge peak in the feedback layers, the superficial ones, whereas the PCC looks more like an inverted U shape. And obviously they also have different numbers of layers and vascular physiology, but these traces and profiles can help us to also say something about hubness. For example, if there's one area that might be actually be the driving force, the hub of the default mode network that's just dragging the other areas passively along with them with feedback input. Another area where you can do layer-dependent fMRI with vaso, for example, would be the primary sensory cortex, just at the other side of the sensory sulcus compared to the motor cortex. And also in the primary sensory cortex, we know the canonical microcircuit, which is actually not so different from the primary visual cortex. Namely, there's input from the thalamus, mostly in the middle layers, and there's cortical feedback input or expectation input, prediction input, or predictive coding feedback in the superficial layers and sometimes secondary in the deeper ones. So we tried to engage these different channels differently with different touching tasks here in collaboration with Yin Hua, where she was in the scanner room while I was scanning outside, and she taught the participants to learn specific sequences of being touched. And the participants always needed to predict which finger is going to be touched next. So they learned the sequence and expect that the index finger will be touched next. However, in some instances, we actually left out the index finger. So for this kind of task, there is no feed-forward input from thalamus. The finger is actually not being touched, only the expectation is there. And for one of the many other control conditions, participants were touched with completely random touching sequences. So here they could not predict which finger is going to be touched next. However, there was input from thalamus. So when you zoom into this tiny part here that represents the index finger in the sensory cortex, you can see that for the feed-forward condition in green, we have this activity in the middle layers, whereas for the expectation input, we mostly have activity in the superficial layers. Better visible also in the group results here in blood volume, where we see this clear superficial peak for vaso and the clear middle peak, feed-forward peak in green for the feed-forward condition. But also in the sensory cortex, it's very beneficial to know where you are across the columnar dimension too. And in the sensory cortex, we have these big, I don't even want to say hypercolumns, but big patches of cortex that refer to the representations of different fingers. So here, for example, in this part, we have the red part that refers to the index finger, and the green part refers to the middle finger. And if we zoom into the index finger area here between the circle and the star, we can see that they are further subdivided into other hypercolumns that are preferentially activated with different frequencies of being touched. These columns that are mostly representing the RA or SA receptors in the skin. And as you can see here, that we do see within the index finger representation that there are these RA receptors columns that kind of span across all the cortical depth, whereas they are interspaced with SA dominated columns that are mostly confined to the middle cortical layers, as expected from the animal literature. So is VASO the best method to do layer-dependent fMRI in humans? I would like to finish this presentation by answering this question again with this bridge metaphor that I showed you earlier. And earlier I was referring to the different spatial regimes of neuroscience, but they can actually also be interpreted as different subdisciplines of neuroscience, where we have on one extreme like the clinical neuroscience, where people do these huge studies of hundreds and thousands of participants. So any kind of imaging methodology that you use over there needs to be very easy. It needs to be cheap, turnkey, non-invasive, whole brain. So VASO is not absolutely perfectly suited for that. Whereas when you do more basic science, like cognitive neuroscience, there you're already getting away with more exotic sequences. Maybe you're even getting away with partial brain coverage. However, any method that you're using here also still needs to be super sensitive to capture these very subtle things that these people care about. For example, they care about the cognitive perception of algebra or algebraic scales or grammar, and those kind of things. Up to all the other side of the bridge, where the researchers are more interested in the chemical, physical processes of brain mechanisms, up to mechanistic systems neuroscience. And there people care more about tuning curves, variations of neurovascular coupling across cortical depth. And there you can get away with all kind of weird exotic sequences. So people do 10 session averages of ASL just to get one question right. Or people even do line scanning, where you throw away 2 dimensions and just do one-dimensional line scanning layer-dependent fMRI. So you can do very weird sequences there. However, in this regime, you're also suddenly in competition with animal research, right? And we have seen earlier from Sung-Lee Kim what kind of amazing methods they have out there, right? And that VASO just cannot compete with that. So while every single discipline of neuroscience here really benefits from layer-dependent fMRI, they differently benefit from VASO layer-dependent fMRI. So if you're interested about the underlying biophysical models of fMRI in animals, then you might be better off with invasive models like these iron contrast agents. And on the other hand, when you do these large population studies and in cases where you do have other means available to account for vascular biases and venous leakage artifacts, there BOLT is more sensitive and might be the way to go. But VASO can really help to bridge the different regimes here. And there are actually a lot of cases where on one hand you need to be non-invasive, and on the other hand you do not really have other means available to account for your vascular biases and venous leakage effects. Actually, in most of the layer-dependent fMRI cases, you do not really have good other means available, I believe. And of course, VASO is not the only method that has this specific sweet spot, but for example, spin echo or 3D grays might fit in somewhere here, or one-dimensional line scanning or 10-hour sessions of ASL might also fit in somewhere here. And each of these methods have their justification and their place. And with this, I thank all my colleagues and I thank you for your attention.